Hello, my name is Siti Nadia binti Sarwan and my metric number is 33DTM19F1076.
Wabarakatuh. My name is Nurhazika Najiha bin Tajuddin and my number matric is 33BTM 19F1078.
Hi, my name is Nurul Nadira binti Saimi. My metric number is 33DTM19F1101.
Hello and today we will present about the topic microscope. Did you know what is microscope? So, introduction of microscope is the compound. Light microscope is a few important laboratory instruments. A good microscope allows us to magnify and observe very small specimens such as bacteria, as well as cell and their various organ cell. All of part of the microscope are important, but without one aspect of a microscope, it will be unable to carry out its basic function. So, if you know, microscope is one thing that important to student using for the experiment and laboratory or found the micro microorganism. So the next one is features of microscope. The number one is complement for scientists research about the medicine, industry, agriculture and education. Second is uniform illumination with consistent color temperature. Third is with excellent flat field aromatical infinity optical system, ensure height, image, flatness across the field. Number 4 is excellent contrast via simple partner iris operation. So number 5 is enable user to remain comfortable during long period of routine microscope observation. So number 6 is the location of the control not maximize ergonomics to improve work efficiency. So this number 7 is magnification. The magnification of microscope is 1000 to 1600 magnification. And so the last one is which camera it can connect to phone or screen or computer. It can also take a photo and a recording video to see the result.
Next is characteristic of microscope. Firstly, it has support at the bottom. Next, it has a lens to adjust distance. Then the third, it is a device used to view a small object. It also has the bottom of the lens like a small glass to place small object. The next characteristic of microscope is the size. It is according to the size set by the laboratory. The color of the microscope is white and black. Then, the last but not least, the price for the microscope at range of the 2,400 ringgit Malaysia.
So the next is body tube. Pieces light from the height to the ears pieces. Next is ears pieces. The ears pieces is where one view the enlarged object. It contains a lens called the ocular that further magnify the specimen by 10 times. Next is arm. Support the lens, mirror and body tube. The microscope should be carried with one hand holding the M and the other under the base. Next is Causes focus keynote. Move the stage up and down quickly. Use it to find a specimen when using the low power objective. Next is fine focus keynote. Use it to make small focus adjustment when using the light power objective. The next is lens source. The lens source illuminate the specimen by shining bright light to it. And the last is power switch. Turn the light on and off.
Okay, for the next function of the microscope is base function. So for the base function is to support the microscope and contains the electronics. The microscope should be carried with one under the base and the other holding the arm. For the diaphragm function, is to rotating dial that controls the passage of light through the stage. Number on the dial indicate the relative amount of light passing, with 5 being the most and 1 being the least. So for the stage function is to hold the slide and contains an opening that allows light to pass through the specimen on its way to the objective. Okay, for the objective lens function, objective produce most of the magnification. The high power lens, which is blue stripe, and magnify 40 times, and the low power lens, which is yellow stripe, magnify 10 times. So the last function for the microscope function is rotating head. The functions is contains mirror and allow the body tube to rotate 360 degrees.
The conclusion in my project is the light microscopy is very powerful tool for understanding the structure and function of tissue, and it is widely used in biomedical science courses as well as in research and diagnostic laboratories. Understanding the capabilities and limitations of the light microscope is important if one is to get the best result from microscopy. The ability to identify structure and specific cell types has been greatly aided by development staining technique and more recently by immunohistochemistry. So the investigation of cell structure has also been considerably extended by the development in electron microscopy and fluorescent microscopy. Thank you.